Dinoflagellate. The dinoflagellates, Greek dinos, whirling, and Latin flagellum, whip, scourge, are monophyletic group of single-celled eukaryotes constituting the phylum Dinoflagellata and usually considered algae. Dinoflagellates are mostly marine plankton, but they also are common in freshwater habitats. Their populations vary with sea surface temperature, salinity, and depth. Many dinoflagellates are photosynthetic, but a large fraction of these are in fact mixotrophic, combining photosynthesis with ingestion of prey, phagotrophy, and myzocytosis. In terms of number of species, dinoflagellates are one of the largest groups of marine eukaryotes, although substantially smaller than diatoms. Some species are endosymbionts of marine animals and play an important part in the biology of coral reefs. Other dinoflagellates are unpigmented predators on other protozoa, and a few forms are parasitic, for example, Oodinium and Pfiesteria. Some dinoflagellates produce resting stages, called dinoflagellate cysts or dinocysts, as part of their life cycles, and is known from 84 of the 350 described freshwater species, and from a little more than 10% of the known marine species. Dinoflagellates are alveolates possessing two flagella, the ancestral condition of the conchs. About 1555 species of free-living marine dinoflagellates are currently described. Another estimate suggests about 2,000 living species, of which more than 1,700 are marine free-living, as well as benthic, and about 220 are from freshwater. The latest estimates suggest a total of 2294 living dinoflagellate species, which includes marine, freshwater, and parasitic dinoflagellates. A rapid accumulation of certain dinoflagellates can result in a visible coloration of the water, colloquially known as red tide, a harmful algal bloom, which can cause shellfish poisoning if humans eat contaminated shellfish. Some dinoflagellates also exhibit bioluminescence, primarily emitting blue-green light. Thus, some parts of the Indian Ocean light up at night, giving blue-green light. Etymology. The term dinoflagellate is a combination of the Greek dinos and the Latin flagellum. Dinos means whirling and signifies the distinctive way in which dinoflagellates were observed to swim. Flagellum means whip, and this refers to their flagella. History. In 1753, the first modern dinoflagellates were described by Henry Baker as animalcules which cause the sparkling light in sea water, and named by Otto Friedrich Müller in 1773. The term derives from the Greek word dinos, meaning whirling, and Latin flagellum, a diminutive term for a whip or scourge. In the 1830s, the German microscopist Christian Gottfried Ehrenberg examined many water and plankton samples and proposed several dinoflagellate genera. These same dinoflagellates were first defined by Otto Büchli in 1885 as the flagellate order Dinoflagellata. Botanists treated them as a division of algae named Pyrophyta or Pyrhophyta, fire algae, Greek pyrhos, fire, after the bioluminescent forms, or Dinophyta. At various times, the cryptomonids, ebriids, and elobiopsids have been included here, but only the last are now considered close relatives. Dinoflagellates have a known ability to transform from non-cyst to cyst-forming strategies, which makes recreating their evolutionary history extremely difficult. Morphology. Dinoflagellates are unicellular and possess two dissimilar flagella arising from the ventral cell side dinocont flagellation. They have a ribbon-like transverse flagellum with multiple waves that beats to the cell's left, and a more conventional one, the longitudinal flagellum, that beats posteriorly. The transverse flagellum is a wavy ribbon in which only the outer edge undulates from base to tip due to the action of the axoneme which runs along it. The axonal edge has simple hairs that can be of varying lengths. The flagellar movement produces forward propulsion and also a turning force. The longitudinal flagellum is relatively conventional in appearance, with few or no hairs. It beats with only one or two periods to its wave. The flagella lie in surface grooves, the transverse one in the cingulum and the longitudinal one in the sulcus, although its distal portion projects freely behind the cell. In dinoflagellate species with dismocant flagellation, e.g. Prorocentrum, the two flagella are differentiated, as in dinoconts, but they are not associated with grooves. Dinoflagellates have a complex cell covering called an amphysema or cortex, composed of a series of membranes, flattened vesicles called alveoli equals amphismal vesicles, and related structures. In thecate armored dinoflagellates, these support overlapping cellulose plates to create a sort of armor called the theca or lorica, as opposed to athecate nude dinoflagellates. These occur in various shapes and arrangements, depending on the species and sometimes on the stage of the dinoflagellate. Conventionally, the term tabulation has been used to refer to this arrangement of thecal plates. The plate configuration can be denoted with the plate formula or tabulation formula. Fibrous extrusums are also found in many forms. A transverse groove, the so-called cingulum or sigillum, runs around the cell, thus dividing it into an anterior episoma and posterior hyposoma. If and only if a theca is present, the parts are called epitheca and hypotheca, respectively. Posteriorly, starting from the transverse groove, there is a longitudinal furrow called the sulcus. The transverse flagellum strikes in the cingulum, the longitudinal flagellum in the sulcus. Together with various other structural and genetic details, this organization indicates a close relationship between the dinoflagellates, the apicomplexa, and ciliates, collectively referred to as the alveolates. Dinoflagellate tabulations can be grouped into 6 tabulation types: gymnodinoid, susoid, goniolochoid, paridinoid. The chloroplasts in most photosynthetic dinoflagellates are bound by 3 membranes, suggesting they were probably derived from some ingested algae. Most photosynthetic species contain chlorophylls in situ, the carotenoid beta-carotene, and a group of xanthophylls that appears to be unique to dinoflagellates, typically peridinin, dinoxanthin, and diadinoxanthin. These pigments give many dinoflagellates their typical golden-brown color. However, the dinoflagellates Carenia brevis, Carenia mycematoi, and Carlidinium micrum have acquired other pigments through endosymbiosis, including fucoxanthin. This suggests their chloroplasts were incorporated by several endosymbiotic events involving already colored or secondarily colorless forms. The discovery of plastids in the apicomplexa has led some to suggest they were inherited from an ancestor common to the two groups, but none of the more basal lines has them. All the same, the dinoflagellate cell consists of the more common organelles, such as rough and smooth endoplasmic reticulum, Golgi apparatus, mitochondria, lipid and starch grains, and food vacuoles. Some have even been found with a light-sensitive organelle, the eye spot or stigma, or a larger nucleus containing a prominent nucleolus. The dinoflagellate erythripsidium has the smallest known eye. Some athecate species have an internal skeleton consisting of 2 star-like siliceous elements that has an unknown function and can be found as microfossils. Tappan gave a survey of dinoflagellates with internal skeletons. This included the first detailed description of the pentasters in Actiniscus pentasterias, based on scanning electron microscopy. They are placed within the order Gymnodiniales, suborder Actinicinae. Theca structure and formation. The formation of thecal plates has been studied in detail through ultrastructural studies. The dinoflagellate nucleus, dinocaryon. Core dinoflagellates, dinokaryotes, have a peculiar form of nucleus called a dinocaryon, in which the chromosomes are attached to the nuclear membrane. These carry a reduced number of histones. In place of histones, dinoflagellate nuclei contain a novel, dominant family of nuclear proteins that appear to be of viral origin, thus are called dinoflagellate viral nucleoproteins DVNPs, which are highly basic, bind DNA with similar affinity to histones, and occur in multiple post-translationally modified forms. Dinoflagellate nuclei remain condensed throughout interphase rather than just during mitosis, which is closed and involves a uniquely extranuclear mitotic spindle. This sort of nucleus was once considered to be an intermediate between the nucleoid region of prokaryotes and the true nuclei of eukaryotes. so were termed mesokaryotic, but now are considered derived rather than primitive traits, i.e. ancestors of dinoflagellates had typical eukaryotic nuclei. In addition to dinokaryotes, DVNPs can be found in a group of basal dinoflagellates known as marine alveolates, mouths that branch as sister to dinokaryotes, syndineals. Classification. Generality. Dinoflagellates are protists and have been classified using both the International Code of Botanical Nomenclature ICBN, now renamed as ICN, and the International Code of Zoological Nomenclature ICSN. About half of living dinoflagellate species are autotrophs possessing chloroplasts and half are non-photosynthesizing heterotrophs. The peridinin dinoflagellates, named after their peridinin plastids, appear to be ancestral for the dinoflagellate lineage. Almost half of all known species have chloroplasts, which are either the original peridinin plastids or new plastids acquired from other lineages of unicellular algae through endosymbiosis. The remaining species have lost their photosynthetic abilities and have adapted to a heterotrophic, parasitic, or kleptoplastic lifestyle. Most, but not all, dinoflagellates have a dinocaryon, described below. See life cycle below. Dinoflagellates with a dinocaryon are classified under Dinokaryota, while dinoflagellates without a dinocaryon are classified under syndineals. Although classified as eukaryotes, the dinoflagellate nuclei are not characteristically eukaryotic, as some of them lack histones and nucleosomes and maintain continually condensed chromosomes during mitosis. The dinoflagellate nucleus was termed mesokaryotic by Dodge 1966 due to its possession of intermediate characteristics between the coiled DNA areas of prokaryotic bacteria and the well-defined eukaryotic nucleus. This group, however, does contain typically eukaryotic organelles such as Golgi bodies, mitochondria, and chloroplasts. Jacob Schiller 1931–1937 provided a description of all the species, both marine and freshwater, known at that time. Later, Elaine Sawerny in 1973, 1978–1982, 1990–1993 listed the new taxonomic entries published after Schiller 1931–1937. Sawerny in 1986 gave descriptions and illustrations of the marine genera of dinoflagellates, excluding information at the species level. The latest index is written by Gomez. Identification. English language taxonomic monographs covering large numbers of species are published for the Gulf of Mexico, the Indian Ocean, the British Isles, the Mediterranean, and the North Sea. The main source for identification of freshwater dinoflagellates is the Susswasser flora. Calcofluor white can be used to stain thecal plates in armored dinoflagellates. Ecology and physiology. Habitats. Dinoflagellates are found in all aquatic environments, marine, brackish, and freshwater, including in snow or ice. They are also common in benthic environments and sea ice. Endosymbionts. All zooxanthellae are dinoflagellates and most of them are members within Symbiodiniaceae, e.g., the genus Symbiodinium. The association between Symbiodinium and reef-building corals is widely known. However, endosymbiontic zooxanthellae inhabit a great number of other invertebrates and protists, for example many sea anemones, jellyfish, nudibranchs, the giant clam Tridacna, and several species of radiolarians and foraminiferans. Many extant dinoflagellates are parasites, here defined as organisms that eat their prey from the inside, i.e. endoparasites, or that remain attached to their prey for longer periods of time, i.e. ectoparasites. They can parasitize animal or protist hosts. Protodinium, Crepidudinium, Piscinudinium, and Blastidinium retain their plastids while feeding on their zooplanktonic or fish hosts. In most parasitic dinoflagellates, the infective stage resembles a typical motile dinoflagellate cell. Nutritional strategies. 3 nutritional strategies are seen in dinoflagellates: phototrophy, mixotrophy, and heterotrophy. Phototrophs can be photoautotrophs or auxotrophs. Mixotrophic dinoflagellates are photosynthetically active, but are also heterotrophic. Facultative mixotrophs, in which autotrophy or heterotrophy is sufficient for nutrition, are classified as amphitrophic. If both forms are required, the organisms are mixotrophic sensu stricto. Some free-living dinoflagellates do not have chloroplasts, but host a phototrophic endosymbiont. A few dinoflagellates may use alien chloroplasts, kleptochloroplasts, obtained from food kleptoplasty. Some dinoflagellates may feed on other organisms as predators or parasites. Food inclusions contain bacteria, blue-green algae, small dinoflagellates, diatoms, ciliates, and other dinoflagellates. Mechanisms of capture and ingestion in dinoflagellates are quite diverse. Several dinoflagellates, both thicate e.g. Serratium hirundinella, Peridinium globulus, and non-thicate e.g. Oxyhis marina, Gymnodinium sp. and Oodinium spp. draw prey to the sulcal region of the cell either via water currents set up by the flagella or via pseudopodial extensions and ingest the prey through the sulcus. In several Protoperidinium spp. e.g. P. canicum, a large feeding veil, a pseudopod called the pallium, is extruded to capture prey which is subsequently digested extracellularly equals pallium feeding. Oblea, Cygibacodinium, and Diplopsalis are the only other dinoflagellate genera known to use this particular feeding mechanism. Catadinium gymnodinium fungiform, commonly found as a contaminant in algal or ciliate cultures, feeds by attaching to its prey and ingesting prey cytoplasm through an extensible peduncle. Two related species, Polycrocos cophoidii and Nitidinium, shoots out a harpoon-like organelle to capture prey. The feeding mechanisms of the oceanic dinoflagellates remain unknown, although pseudopodial extensions were observed in Podolampus bites. Blooms. Introduction. Dinoflagellate blooms are generally unpredictable, short, with low species diversity, and with little species succession. The low species diversity can be due to multiple factors. One way a lack of diversity may occur in a bloom is through a reduction in predation and a decreased competition. The first may be achieved by having predators reject the dinoflagellate by, for example, decreasing the amount of food it can eat. This additionally helps prevent a future increase in predation pressure by cause predators that reject it to lack the energy to breed. A species can then inhibit the growth of its competitors, thus achieving dominance. Harmful algal blooms. Dinoflagellates sometimes bloom in concentrations of more than 1 million cells per milliliter. Under such circumstances, they can produce toxins generally called dinotoxins in quantities capable of killing fish and accumulating in filter feeders such as shellfish, which in turn may be passed on to people who eat them. This phenomenon is called a red tide, from the color the bloom imparts to the water. Some colorless dinoflagellates may also form toxic blooms, such as Pfiesteria. Some dinoflagellate blooms are not dangerous. Bluish flickers visible in ocean water at night often come from blooms of bioluminescent dinoflagellates, which emit short flashes of light when disturbed. A red tide occurs because dinoflagellates are able to reproduce rapidly and copiously as a result of the abundant nutrients in the water. Although the resulting red waves are an interesting visual phenomenon, they contain toxins that not only affect all marine life in the ocean, but the people who consume them as well. A specific carrier is shellfish. This can introduce both non-fatal and fatal illnesses. One such poison is saxitoxin, a powerful paralytic neurotoxin. Human inputs of phosphate further encourage these red tides, so strong interest exists in learning more about dinoflagellates from both medical and economic perspectives. Dinoflagellates are known to be particularly capable of scavenging dissolved organic phosphorus for P nutrient. Several species have been found to be highly versatile and mechanistically diversified in utilizing different types of DOP. The ecology of harmful algal blooms is extensively studied. Bioluminescence. At night, Water can have an appearance of sparkling light due to the bioluminescence of dinoflagellates. More than 18 genera of dinoflagellates are bioluminescent, and the majority of them emit a blue-green light. These species contain scintillans, individual cytoplasmic bodies about 0.5 in diameter, distributed mainly in the cortical region of the cell, out pockets of the main cell vacuole. They contain dinoflagellate luciferase, the main enzyme involved in dinoflagellate bioluminescence, and luciferin, a chlorophyll-derived tetrapyryl ring that acts as the substrate to the light-producing reaction. The luminescence occurs as a brief 0.1 sec blue flash max 476 nm when stimulated, usually by mechanical disturbance. Therefore, when mechanically stimulated by boat, swimming, or waves, for example, a blue sparkling light can be seen emanating from the sea surface. Dinoflagellate bioluminescence is controlled by a circadian clock and only occurs at night. Luminescent and non-luminescent strains can occur in the same species. The number of scintillans is higher during night than during day, and breaks down during the end of the night at the time of maximal bioluminescence. The luciferin luciferase reaction responsible for the bioluminescence is pH sensitive. When the pH drops, luciferase changes its shape, allowing luciferin, more specifically tetrapyryl, to bind. Dinoflagellates can use bioluminescence as a defense mechanism. They can startle their predators by their flashing light, or they can ward off potential predators by an indirect effect such as the burglar alarm. The bioluminescence attracts attention to the dinoflagellate and its attacker, making the predator more vulnerable to predation from higher trophic levels. Bioluminescent dinoflagellate ecosystem bays are among the rarest and most fragile, with the most famous ones being the bioluminescent bay in La Pargara, Lodges, Puerto Rico, Mosquito Bay in Vieques, Puerto Rico, and Los Cabezas de San Juan Reserva Natural Fajardo, Puerto Rico. Also, a bioluminescent lagoon is near Montego Bay, Jamaica, and bioluminescent harbors surround Castine, Maine. Within the United States, Central Florida is home to the Indian River Lagoon, which is abundant with dinoflagellates in the summer and bioluminescent C10 in the winter. Lipid-sterol production. Dinoflagellates produce characteristic lipids and sterols. One of these sterols is typical of dinoflagellates and is called dinosterol. Transport. Dinoflagellate feca can sink rapidly to the seafloor in marine snow. Life cycle. Cycle. Introduction. Dinoflagellates have a haplontic life cycle, with the possible exception of Noctiluca and its relatives. The life cycle usually involves asexual reproduction by means of mitosis, either through dysmostases or eleuterostases. More complex life cycles occur, more particularly with parasitic dinoflagellates. Sexual reproduction also occurs, though this mode of reproduction is only known in a small percentage of dinoflagellates. This takes place by fusion of two individuals to form a zygote, which may remain mobile in typical dinoflagellate fashion and is then called a planozygote. This zygote may later form a resting stage or hypnozygote, which is called a dinoflagellate cyst or dinocyst. After or before germination of the cyst, the hatchling undergoes meiosis to produce new haploid cells. Dinoflagellates appear to be capable of carrying out several DNA repair processes that can deal with different types of DNA damage. Dinoflagellate cysts. The life cycle of many dinoflagellates includes at least one non-flagellated benthic stage as a cyst. Different types of dinoflagellate cysts are mainly defined based on morphological number and type of layers in the cell wall and functional long or short-term endurance differences. These characteristics were initially thought to clearly distinguish pellicle thin-walled cysts from resting double-walled dinoflagellate cysts. The former were considered short-term temporal, and the latter long-term resting cysts. However, during the last 2 decades, further knowledge has highlighted the great intricacy of dinoflagellate life histories. More than 10% of the approximately 2,000 known marine dinoflagellate species produce cysts as part of their life cycle. See diagram on the right. These benthic phases play an important role in the ecology of the species, as part of a planktonic benthic link in which the cysts remain in the sediment layer during conditions unfavorable for vegetative growth and from there re-inoculate the water column when favorable conditions are restored. Indeed, during dinoflagellate evolution, the need to adapt to fluctuating environments and/or to seasonality is thought to have driven the development of this life cycle stage. Most protists form dormant cysts in order to withstand starvation and UV damage. However, there are enormous differences in the main phenotypic, physiological, and resistance properties of each dinoflagellate species cysts. Unlike in higher plants, most of this variability, for example in dormancy periods, has not been proven yet to be attributed to latitude, adaptation, or to depend on other life cycle traits. Thus, despite recent advances in the understanding of the life histories of many dinoflagellate species, including the role of cyst stages, many gaps remain in knowledge about their origin and functionality. Recognition of the capacity of dinoflagellates to encyst dates back to the early 20th century in biostratigraphic studies of fossil dinoflagellate cysts. Paul Range was the first to identify cysts as the fossilized remains of dinoflagellates. Later, cyst formation from gamete fusion was reported, which led to the conclusion that encystment is associated with sexual reproduction. These observations also gave credence to the idea that microbial encystment is essentially a process whereby zygotes prepare themselves for a dormant period. Because the resting cysts studied until that time came from sexual processes, dormancy was associated with sexuality, a presumption that was maintained for many years. This attribution was coincident with evolutionary theories about the origin of eukaryotic cell fusion and sexuality, which postulated advantages for species with diploid resting stages in their ability to withstand nutrient stress and mutational UV radiation through recombinational repair, and for those with haploid vegetative stages as asexual division. Nonetheless, certain environmental conditions may limit the advantages of recombination and sexuality, such that in fungi, for example, complex combinations of haploid and diploid cycles have evolved that include asexual and sexual resting stages. However, in the general life cycle of cyst-producing dinoflagellates as outlined in the 1960s and 1970s, resting cysts were assumed to be the fate of sexuality, which itself was regarded as a response to stress or unfavorable conditions. Sexuality involves the fusion of haploid gametes from motile planktonic vegetative stages. Endogenously, a species-specific physiological maturation minimum period dormancy is mandatory before germination can occur. Thus, hypnozygotes were also referred. Exogenously, germination is only possible within a window of favorable environmental conditions. Yet, with the discovery that planozygotes were also able to divide, it became apparent that the complexity of dinoflagellate life cycles was greater than originally thought. Following corroboration of this behavior, in several species, the capacity of dinoflagellate sexual phases to restore the vegetative phase, bypassing cyst formation, became well accepted. Further, in 2006 Kremp and Paro showed the dormant resting cysts of the Baltic cold-water dinoflagellates, Scripsiella hangoi and Gymnodinium sp. were formed by the direct encystment of haploid vegetative cells, i.e. asexually. In addition, for the zygotic cysts of Pfiesteria piscicida dormancy was not essential. Genomics. One of the most striking features of dinoflagellates is the large amount of cellular DNA that they contain. Most eukaryotic algae contain on average about 0.54 pgDNA/cell, whereas estimates of dinoflagellate DNA content range from 3,250 pg/cell, corresponding to roughly 3,215 MBA. In comparison, the haploid human genome is 3,180 MBA, and polyploidy or polytony may account for this large cellular DNA content, but earlier studies of DNA reassociation kinetics and recent genome analyses do not support this hypothesis. Rather, this has been attributed, hypothetically, to the rampant retroposition found in dinoflagellate genomes. In addition to their disproportionately large genomes, dinoflagellate nuclei are unique in their morphology, regulation, and composition. Their DNA is so tightly packed that exactly how many chromosomes they have is still uncertain. The dinoflagellates share an unusual mitochondrial genome organization with their relatives, the apicomplexa. Both groups have very reduced mitochondrial genomes around 6 kilobases kb in the apicomplexa ds-16 for human mitochondria. One species, Amoebophria ceratiae, has lost its mitochondrial genome completely, yet still has functional mitochondria. The genes on the dinoflagellate genomes have undergone a number of reorganizations, including massive genome amplification and recombination, which have resulted in multiple copies of each gene and gene fragments linked in numerous combinations. Loss of the standard stop codons, trans-splicing of mRNA for the mRNA of COX-3, and extensive RNA editing recoding of most genes has occurred. The reasons for this transformation are unknown. In a small group of dinoflagellates, called diatoms durinschia and cryptoperdinium, the endosymbionts diatoms still have mitochondria, making them the only organisms with two evolutionarily distinct mitochondria. In most of the species, the plastid genome consists of just 14 genes. The DNA of the plastid in the peridinin-containing dinoflagellates is contained in a series of small circles. Each circle contains one or two polypeptide genes. The genes for these polypeptides are chloroplast-specific because their homologs from other photosynthetic eukaryotes are exclusively encoded in the chloroplast genome. Within each circle is a distinguishable core region. Genes are always in the same orientation with respect to this core region. In terms of DNA barcoding, its sequences can be used to identify species, where a genetic distance of p equals 0.4 can be used to delimit species, which has been successfully applied to resolve long-standing taxonomic confusion, as in the case of resolving the Alexandrium tamarense complex into 5 species. A recent study revealed a substantial proportion of dinoflagellate genes encode for unknown functions, and that these genes could be conserved and lineage-specific. Evolutionary history. Dinoflagellates are mainly represented as fossils by fossil dinocysts, which have a long geological record with lowest occurrences during the mid-Triassic, whilst geochemical markers suggest a presence to the early Cambrian. Some evidence indicates dinosteroids in many Paleozoic and Precambrian rocks might be the product of ancestral dinoflagellates, protodinoflagellates. Molecular phylogenetics show that dinoflagellates are grouped with ciliates and apicomplexans equals Thorizo in a well-supported clade, the alveolates. The closest relatives to dinokaryotic dinoflagellates appear to be apicomplexans, Perkinsis, Parvolucifera, Syndinians, and Oxyanes. Molecular phylogenies are similar to phylogenies based on morphology. The earliest stages of dinoflagellate evolution appear to be dominated by parasitic lineages, such as Perkinsids and Syndinians, e.g. Amoebophria and Hematidinium. All dinoflagellates contain red algal plastids, or remnant non-photosynthetic organelles of red algal origin. The parasitic dinoflagellate Hematidinium, however, lacks a plastid entirely. Some groups that have lost the photosynthetic properties of their original red algae plastids has obtained new photosynthetic plastids, chloroplasts, through so-called serial endosymbiosis, both secondary and tertiary. Like their original plastids, the new chloroplasts in these groups can be traced back to red algae, except from those in the members of the genus Lepidodinium, which possess plastids derived from green algae, possibly Trebouxia or Ulvaphyceae. Lineages with tertiary endosymbiosis are Dinophysis, with plastids from a cryptomonad, Vicarinia, Carlodinium, and Takayama, which possess plastids of haptophyte origin, and the Peridiniaceae, Durinschia and Cryptoperdinium, which has plastids derived from diatoms. Some species also perform kleptoplasty. Dinoflagellate evolution has been summarized into 5 principal organizational types: prorocentroid, dinophysoid, goniolacoid, peridinoid, and gymnodinoid. The transitions of marine species into freshwater have been infrequent events during the diversification of dinoflagellates and in most cases have not occurred recently, possibly as late as the Cretaceous. Recently, the living fossil Oodinium and Pfiesteria was found inhabiting the Indo-Pacific warm pool, which served as a refugium for thermophilic dinoflagellates. Examples: Alexandrium, Goniolax, Gymnodinium, Lingulodinium polyedrum.